Welcome, everyone. Welcome to day 3 of Fatty Acid Symposium, our first annual symposium presented by the Fatty Acid Research Institute and OmegaQuant. We want to thank our co-partners, FARI, Fatty Acid Research Institute, Carlson Labs, Nordic Naturals, Yoli, Vivo Omega, Nutraceuticals World, OmegaQuant, and GoEd. So thank all of our co-partners. There are no sponsors for this event, strictly co-partners that share our information with all of their healthcare providers and researchers throughout. Let's go ahead and get started. I want to introduce Dr. Harris. This is a topic, right? Most people want to live longer. And there's a lot of things in the world right now around lifespan. There's a few people that are trying to hack things, and they say they can live to 150, 200. So where does omega-3 fit into this? And that's what Dr. Harris is going to talk. He's the president of the Fatty Acid Research Institute. He's been leading researcher in the omega-3 fatty acid field for over 40 years, more than 360 scientific papers on fatty acids and health, with the vast majority on omega-3. He's been on faculty of 3 medical schools at the universities of Kansas, Missouri, at Kansas City, and South Dakota. He's received 8 NIH grants to study omega-3 and was also the co-author on 3 American Heart Association statements on fatty acids and heart health. Is also the co-inventor of the omega-3 index test and other blood tests, and founder of OmegaQuant Analytics. Dr. Harris has been ranked among the top 2% of the scientists worldwide for his impact on the research. Doc, thank you so much for being here, and I'm going to turn it over to you.
Great. Thank you, Tavis. Good to be back. Welcome, everybody. Hope this series of seminars is being helpful for you. Obviously, I'm going to talk about longevity, and we'll really run quite a span of types of studies and evidence that we'll look at that pool together. Ecological means basically looking country versus country. A little bit of animal study I'll go into, and we'll then get into human studies, observational studies, and some that are based on what dietary intake estimates of omega-3 and longevity, and then others that are biomarker blood omega-3 levels based studies in that category. A little bit of work on interventional studies, meaning the effects of actually giving omega-3 supplements on longevity, and then have a quick look at mechanisms. So that's where we're going today. We'll just remind everybody, it's a blood test that we pioneered about 20 years ago. It's the amount of EPA and DHA in red cell membranes, expressed as a percent. This particular cartoon, you can see there's 64 fatty acids in this little cutout of the red cell membrane, and 4 of them are omega-3. That gives you an omega-3 index of 6.25%. That's the way it works. Pretty straightforward. More omega-3 in your diet, more in your blood. The scale we're looking at, we think desirable is 8% to 12% omega-3, which is typical for Japan, as we'll see in a moment. Typical for Europe and the U.S. is somewhere down here in the intermediate zone. And again, that's try to keep that 8 to 12% value in mind. So back to ecological data. Set the stage here for, I mentioned omega-3 is high in Japan. These are data published 2008, looking at Japanese individuals, men and women, by decade of life. And so across the bottom, different in people in their 20s, 30s, 40s, 50s, and 60s. As you can see, the omega-3 index is what's measured here. It goes up with age, probably driven a lot by dietary habits. It goes up for men and for women. Women are in blue, men are in black. And you have a comparison here with the average level in omega-3 index in 60-year-old people in Boston area is about half of what it is in Japanese. But there's the 8%. Roughly Japanese, you could say, have an omega-3 index around 8%. Just across the water from them is Korea. And we have a researcher in our group at FARI, Dr. Young-Soon Park, who has published many studies looking at the omega-3 index in Korea. And this is kind of a quick summary. You can see from all these different cohorts she's looked at, the omega-3 index is at least 8, if not higher, in Korea. So here are 2 countries where fish is consumed quite readily, and they have much higher omega-3 levels. Well, it's like, so what? Okay, well, part of the so what, and this is the ecological stuff. Here we'll just look at death rate from coronary heart disease across these 3 countries. In the U.S., the recent data I had here was a few years ago, hadn't changed much. 87 per 100,000 people die of heart disease. Okay. In South Korea, it's 21 people. In Japan, it's 28 people. So almost 20 or 75% less people, less death from coronary heart disease, which, since it's a major killer, it's going to result in longer life. And to that point, let's compare Japan, South Korea, and the US in terms of longevity. This is longevity at birth. And Japan is number 2 in the world at 84.5 years is the life expectancy. I think this is for men, probably. Men and, yeah, well, it could be for women. Or actually it could be for the average of both. But in any event, 84.5 years, number 2. South Korea is sitting at number 9, which is 82.8 years. And the United States sits at number 35 at just around 79 years. So almost a 5-year difference in total mortality and longevity. Now, of course, this is ecological data, meaning this countries by countries. You cannot draw any conclusions other than the fact that this is consistent with the rest of the story we're going to say. But we can't say the omega-3s are causing this, but they may be involved. Anyway, it's part of the study. One study on, there have been other studies on mice, but this is the effects of giving omega-3 to mice who have autoimmune disease. They have a certain model. And if they carry fish oil is given to some, we're looking at the control oil and ad lib fish oil, ad lib diets. The ad lib means that mice are able to eat whatever they want. FR is food restriction, which we know extends lifespan. But if you look at the survival of mice beginning at the study, that's the death rate of mice that are on the control diet. This is the death rate of the mice that are on the fish oil diet. If you calorie restrict them and then look at the omega-3, the calorie restriction increases lifespan, and the omega-3 added to it increases it even more. So there was certainly a relationship. There have been other studies where you have not seen an effect of omega-3 in certain mouse models, but I don't want to go into all that. I'll just simply say there's some evidence in mice for this, and there's some that doesn't support it. Let's get over to humans. We're going to talk about the relationship in this study between estimated dietary omega-3 intake and the rate of what's called phenotypic age acceleration. So we're looking at, we'll see quite a bit here about calculating biologic age versus chronological age. Chronological age is very easy. You look at the calendar and you know how many years you've been alive. Biologic age is kind of a health age. Are you healthier than the average person is at 60, at 50, or are you less healthy than the average person? So that's your own biologic age, and biologic and chronologic age can differ. And so in this particular study, quoted here from the abstract, the purpose of the study was to investigate the association between dietary omega-3 fatty acid intake. And I will say this, when they say dietary intake, they mean all omega-3s, including ALA. So I'm disappointed that they didn't give data just for EPA and DHA, but they're covering all omega-3s. And what they call accelerated phenotypic aging. Phenotypic is just the way your body is. You're the phenotype, not as opposed to a genotype. So accelerated aging, don't worry about the word phenotypic. It's referred to as the PhenoAge XL metric. We'll look at that in a minute. PhenoAge XL is defined as the difference between your phenotypic or your biologic age, calculated using blood biochemical markers, and your chronological age. So they use data from the NHANES cohort, 1999 to 2018, 20,000 individuals, cross-sectional study. And we'll see, they calculated this PhenoAge XL metric on all 20,000 people, and they estimated omega-3 intake. And we'll start with, if you would like to calculate your PhenoAge, well, what could be easier? Here's the equation over here, and we are not going to go through this, but it is, see this XB variable here is sitting up here put the exponent upon the exponent, and then gamma does something, and these are all the different things they measure in the blood that give them an idea of your actual biologic age. Very, I mean, piece of cake to do this, obviously. And so what did they find? Well, they found if you look at for men, there's actually a difference between men and women. Along the bottom axis is the reported intake of omega-3 fatty acids in grams per day. And what they showed was if your intake is above roughly a couple of grams here, roughly, if that's the inflection point, then your biologic age is less than your chronologic age. You're younger than your years say. That's what the blue line is. If your omega-3 intake is less, then your actual biologic age is greater than your chronologic age. So, of course, where we all want to be is down in this area. So this showed that there was a relationship between the amount of omega-3 consumed and this calculator that produced this metric, their pheno age. For women, it's not as dramatic. The same pattern is there. It's just not quite as dramatic as it is for men. And so what they concluded here, increased omega-3 intake associated with slowdown in the pheno age accelerator, especially in men. The threshold effect identifies an intake of around 1 gram. I would say probably maybe more like 2 or 2.5 grams on this particular metric. And so higher omega-3, point taken from this is a higher omega-3 is important. They concluded the study highlights the potential role of dietary omega-3 fatty acids in regulating pheno age XL, your biologic age, and supports the strategy of delaying the aging process through dietary interventions to increase omega-3. So they did a great job. I like their conclusions. This was a 2024 study, so we're looking at new stuff. We can look back at some older data, 10 years ago. This is data that was collected, look down here, on about 70,000 individuals living in the state of Washington. Were followed for about 5 years from baseline until death from any cause, when there were about 3,000 deaths out of that 70,000. And they had data on these individuals that from which they could calculate the EPA and DHA intake from both diet and supplements in grams per day. And so this was sort of the quartiles of omega-3, long-chain omega-3 intake. These are your hazard ratios. And this is the trend, I think, to highlight this, 0.86. This is a 14% reduction in risk for death over that 5 years, and an intake of EPA and DHA over 3.22 milligrams per day. That's what this value is here. So that's simply points to the fact that the higher the intake, the lower the risk for death. The results suggested that the intake of omega-3 may reduce risk for total mortality and cancer specific, but we didn't talk about that part. So this study, again, looking at reported intake of EPA and DHA, not blood levels, but intake, linked higher omega-3 levels with greater lifespan. Lower risk for death is the other side of the coin of greater longevity. How about omega-3 supplements? These are data from the UK Biobank, a study that included about 428,000 people who reported on their habitual use of fish oil supplements, yes or no. Do you take supplements or not, without specifying how much or anything. And they're looking at a variety of outcomes. Total mortality over around 10 years is the primary one I'm interested in. And so here is the actual number of people that died who are the non-users, 8781, fish oil users, 4147, and they did the statistical models, and they found here what would be right here for total mortality, 13%, was 14% in the other paper, 13% lower risk for death for fish oil supplement users in this particular study. Now, that's not the optimal way to do it. Just asking people to take fish oil is pretty poor man's way of getting it at the blood levels of omega-3. So we did some studies, and I presented this on Monday, so we'll go through it quickly. But we did a study pooling 17 cohorts of observational cohorts on which omega-3 levels in the blood were known. And we asked the question, is there a relationship between blood omega-3 and risk for death? And here we'll look at four different groups. This group on the far left, these 5 columns reflect the quintiles of omega-3 levels in the blood across all these 17 cohorts. And the dark blue bar is the reference group, the lowest quintile, the lowest omega-3 level. And as the bars go higher and higher, or up and up, the risk for death drops in a very monotonic way, which is one of the strongest indications for causality, that there's a relationship that's linear, to the point where the people at the highest omega-3 quintile, highest 25%, 20%, were about 20% less likely to die over the study period. And that's to all-cause mortality. We also saw cardiovascular disease mortality, cancer mortality, and all other causes combined, mortality was reduced with omega-3. To give you some sense of what a quintile 1 and a quintile 5 omega-3 index are, roughly 3.6 at the low end, 7.8 at the top end, giving some good support for the idea that an 8% target is a very reasonable one. What I didn't show on Monday is essentially a strong confirmation of the study I just showed you. This is done just in the UK Biobank, where we looked at plasma DHA levels, which is what they have available. They didn't do red blood cell, and they didn't do omega-3 index. But the plasma DHA is pretty strongly correlated with the omega-3 index. And this study, we did exactly the same thing. We looked at quintiles of DHA levels and risk for all-cause mortality here. And this, you can see here again, going from Q1 to Q5, quintile 1 to quintile 5, the risk for death dropped by about 20% at the far end. CVD mortality, the same pattern. Cancer mortality, same pattern. Other mortality, also the same pattern. So we strongly confirmed what we had seen in the 17 cohort study with one very large cohort. This included data from about 215,000 people. In the UK Biobank, we estimate that the quintile 1 omega-3 level was roughly about 3.8%, consistent with the previous one, and quintile 5 was over 8%. So again, confirming that 8% target makes some sense. So those are 2 big studies that have shown omega-3. I'll show you maybe one more here. This is the LURIC study. This was a study done in Germany. 3,200 patients who were undergoing diagnostic catheterization had their blood drawn, and then they were followed up over the next 10 years for risk for death. And we got data on the quintiles of omega-3 and tertiles, thirds. Lowest tertile had an omega-3 index of 4.5. The highest had an omega-3 index average of 7.3. And there was a 22% lower risk for death across that spectrum. So time and time again, we see the same thing. Higher omega-3 levels, lower risk for death. This is a paper from Heidi Lai, who's now one of our FARI scientists. This is when she was working with Dr. Mozaffarian in Boston. And they were looking at the question of unhealthy aging, because there's, of course, the opposite of that is healthy aging, but they're looking at risk for unhealthy aging. It's hard to talk about risk for healthy aging because that's not something you're at risk for, but you are at risk for unhealthy aging. So what did they do? They looked in one particular cohort, the cardiovascular health study. This is the blood levels of omega-3 done in quintiles, so groups of 5. And here's the median fatty acid levels in the blood. You see, this is plasma phospholipid, so this is not red blood cell levels. And hazard ratios, meaning what's the risk relative to group 1. And they showed here as the omega-3 levels went up, the risk for unhealthy aging went down. And so 0.82 is the hazard ratio. That means the risk for unhealthy aging was 18% lower compared to in the highest versus the lowest omega-3 level. And what do they mean by unhealthy aging? It's reaching age 65 or rather developing any of the following diseases after age 65. If you don't, you're aging healthily: CVD, cancer, lung disease, severe chronic kidney disease, cognitive or physical dysfunction, or death. So if you don't develop those things in a 9-year period after you turn 65, you're considered to have healthily or aged healthily. If you got any one of those, you had unhealthy aging. And so higher omega-3 levels associated with lower risk for that. Moving to a study we talked about on Monday. This is a study headed up by another one of our FARI scientists, Dr. Michael McBurney. And we were working in the Framingham cohort. We were looking for a red blood cell fatty acid fingerprint to predict risk for all-cause mortality. So more than just the omega-3 index, for example. And we talked about this finding in the study where having a high omega-3 was sort of like was kind of equivalent to being a non-smoker in terms of your long-term total mortality in time. So I don't want to go over that one again. I do want to look at this. We also calculated in that paper a theoretical impact on lifespan of different levels of the omega-3. So what are the effects of having an omega-3 index that's 1, 2, 3, 4 quintiles higher than Q1 on theoretical risk for death? So we developed a model to predict risk for death. And I wanted to show, first of all, how the model performs on things that we know make sense. So for example, in this statistical model that we're going to be looking in the next slide for omega-3, in the same one, being a woman versus being a man added 3.6 years of life, which is just about right. Women live about 3.5, 4 years longer than men. So the model captured that properly. Being a non-smoker versus being a smoker adds 4.7 years of life. That's strong. And again, that makes sense. The model is working. Not having diabetes versus having diabetes adds about 3.9 years of life. So this was also predicted from the model, and that fits with reality. So to the extent that we can trust this model to actually predict effects on lifespan, here's what we saw for omega-3. So if you had if your omega-3 index was in Q2, the second quintile compared to Q1, you lived about 1.18 years longer than people in Q1. If your omega-3 index was in Q3, quintile 3, compared to quintile 1, 2.36 years. If you go up from the lowest quintile to the highest quintile, quintile 5, that added about 4.72 years of life. So if you want to get some kind of idea, again, here are the estimated omega-3 index values for Q1 and Q5, just to give you an idea. The point is here that we're looking at serious differences in lifespan. We're not talking about a month or two. This is years as a function of how much omega-3 you've got in your blood. Now, this doesn't prove that the omega-3 is causing it, but it's there. Where there's smoke, there's fire. And we think this is a compelling evidence that the omega-3 index really is a predictor of these kinds of things. Golly, about 15 years ago, we participated in a study called the Heart and Soul Study out in San Francisco, which was done with individuals who had about 1,000 people who had stable coronary heart disease. And we measured the omega-3 index, and then we divided them basically into two groups, those above and below the median value of the omega-3, 5.4 versus above and below. And we found the people who had the higher omega-3 index were on the top curve. This is the mortality curve, survival probability. They had a higher probability of surviving out that 10 years. And it took roughly about 1.2 years difference between the time when 20% of this group versus 20% of that group died. So again, roughly in this case, about 1.2 years. That's not as easy to interpret, I think, perhaps, as the previous data we had from Framingham. But here's a completely different cohort. We're seeing exactly the same thing. And to explain some of this, we need to talk a little bit about telomeres. And I think many of you know about telomeres and how they are, the rate at which they shrink is some kind of a measure of biologic aging. So just to get you up to speed on that, when we talk about telomeric aging or telomere attrition. What we're talking about is these little tips on the ends of the chromosomes in every cell in your body. These gray parts are called the telomeres. Telo means the end in Greek. And what happens is every time a cell divides, here's the mother cell, and she divides into two daughter cells. All of the DNA from the mother gets into the daughter, except for a little bit of the tip of the telomere. Some of it just doesn't make it. When the daughter cells divide into the granddaughter cells, a little bit more of the telomere is gone. And et cetera, generation after generation, until there is no telomere left. At this point, the cells can't divide anymore and the cells will die. So if you can get some idea of how fast these telomeres are shrinking, we call that telomere attrition, you get some idea of how fast someone is aging, because as again, your cells are turning over faster, you're probably aging faster. And so back to the heart and soul study where we just looked at that data, one of the investigators there looked at the omega-3 level as a predictor of the rate of telomere attrition in this study. And what they found was the patients with the highest omega-3 index experienced the slowest rate of telomere shortening, or telomere attrition, or we'll call it cellular aging. One standard deviation increase in the omega-3 index was associated with 32% reduction in the odds of telomere shortening. If we look over here in this right box, this is along the bottom axis is 4 categories of the omega-3 index, or quartiles, and from low omega-3 index to high omega-3 index. And this dot here represents how much shrinkage the telomeres experienced in people with low omega-3 versus high omega-3. And you can see a very strong dose-related relationship. The higher the omega-3 index, the less shrinkage, less biologic age accumulation was seen in this study. And so that fits with the idea that the omega-3s actually are keeping the body healthy enough so the cells don't live longer, they don't have to reproduce as often, which slows the rate of telomere attrition. We can look now at a study from a couple of years ago. Again, it was a study done within the UK Biobank. And this was a very wide-ranging study. It looked at modifiable factors associated with leukocytes. So leukocytes are white blood cells. That's where they typically measure the telomere, the length. They get the DNA out of the white blood cells. And they're looking at telomere length. And the idea is there must be some factors associated with having shorter telomeres and some factors associated with having long telomeres. And what they found, I keep hitting the wrong button. Sorry about that. They found 17 factors that were statistically significantly related to the length of telomeres in this cohort, which was, and this was probably the full, no, this one might have been half the cohort, like 250,000 people. No, can't remember exactly. It was a lot of people. And so in multivariable analysis of 17 traits with strong associations with telomere length, and strong meant equivalent to 2 or more years of age-related change in leukocyte telomere length. So again, shorter telomeres is older age, is what it amounts to. So in the multivariable analysis of these 17 traits, only 3 of them held up to have statistically significant relationships. And one of them was oily fish intake. One was educational attainment. And the third one was general health status, which is a pretty vague thing. But the thing you can really do something about is oily fish intake or omega-3 intake. And so show here, these are those 17 factors. Here, if it's over on this side of the dotted line, you have longer telomeres associated with oily fish intake, and that's out here. The 3 things that they mentioned: poor overall health, was negative effects. So that's what they're talking about, general health status. That factor was statistically significant in multivariable analysis. The omega-3 or the fish intake and the educational Where's educational? So yeah, university college degree. That was another factor. But the ones that you can really do something about in any given, just in your normal life, is you can change your omega-3 status. So this was evidence from the UK Biobank that at least they didn't measure omega-3 in this particular study. They just looked at reported intake of oily fish. Now we can look a little bit at randomized trials now, because this is all observational. But there is some good evidence, not nearly as much, but there's good evidence that omega-3 supplementation will raise telomerase levels. The telomerase is an enzyme in your white blood cells or in your cells that actually increases the length of a telomere. So normal cell division, you lose telomeres every time you divide. But if you've got a more active telomerase, the cell can actually extend or add back or increase the length of the telomere, which would, again, allow the cell to keep reproducing longer. So in this particular study, so you want to have a high telomerase activity. That's a good thing. So in this study, with just 71 patients with schizophrenia who had been given 2.2 grams of EPA and DHA or placebo for about 26 weeks, so about a half a year. So, and the investigators looked at the effects on the activity, enzyme levels of telomerase. And so what they found here was a baseline. This is the telomerase activity, telomerase levels, the activity, or I keep saying activity, the enzyme levels in the blood, in the cells. And at baseline, there was no difference. The group that got omega-3, over that 2 or 6 month period, the level of telomerase went up compared to placebo. So taking omega-3 was associated with increased synthesis of this enzyme that allows your telomeres to stay long. So that's a good effect. How about randomized trials where we just looked at the risk for death? That's the most simple study, and I will pick this one out from Rizzo's et al. And from 2021. This is a meta-analysis of the effects of EPA and DHA on a variety of outcomes, cardiovascular outcomes, but also total mortality. So this is data from 17 randomized trials of greater than 1 year duration, about 83,000 people. And they were looking at doses, the effects of different doses. And so here's a 0.84 grams a day or less. That's one capsule of Leveza, Omicor, or less. Oh, excuse me, less than that. One full, these are the little capsule pictures they use. One full capsule, that's this dose, and then over 2.5 grams is the 3 capsules plus. And so if you look up here in the total mortality area, they looked at, again, a half pill, 1 pill, 2, and 3 or more pills. There was certainly a trend toward less total mortality, although these 3 studies didn't quite statistically significantly lower it, but relative risk was 0.11. However, if you ask the question, what if you pooled all these studies, 5 studies, including that one and that one, then you get a statistically significant reduction in risk for death if you look at those studies together associated with fish oil supplement use. And so that's some randomized trial data that fish oil supplement use does reduce risk of total mortality. The same can be seen in the Reduce It study. This is the Reduce It USA cohort, which is about 3,100 patients that were given even a mineral oil placebo or Vascepa, the EPA ethyl ester product, for about 5 years, 4 grams a day of each. And of the many outcomes that they reported, the last one here is total mortality. And they found a statistically significant 30% reduction in risk for death from any cause in the group that got the omega-3 fatty acids. So that is randomized trial data that omega-3s do reduce risk for death. Very interesting paper, just published, actually published on February 3rd. So what is today? Like February 8th or 9th or 10th or something. So I was pleased to see this come out. This is a paper in one of the Nature journals. individual and additive effects of vitamin D, omega-3, and exercise on DNA methylation clocks. So now we're also looking at these clocks. Many people are developing aging clocks that are based on DNA, and to what extent the DNA has been methylated. The more methylation, typically, the faster you're aging. But they're methylation clocks, a biological aging in older adults from the Do Health trials. The Do Health was a randomized trial where people were randomized to either vitamin D, omega-3, or exercise, or some combination of both, like 9 different treatment groups. And the general idea that we're looking at here, this is not from that paper, but this is from another paper. Take for example, 2 baby girls born at year 0, and they're growing up. The blue one here they're both developing normally at age 20, but all of a sudden here at age 40, the blue one is a little bit thinner, a little bit healthier. The brown gal is adding weight, getting a little more intact, and eventually and getting older, and by age, say, 68 is dead, as opposed to this one who is aging slower and lives longer. So, of course, they are both they've still aged at the same rate chronologically, but this one, the blue one, is aging at a slower rate biologically. And so the idea of these clocks is to be able to predict or detect people who are aging more slowly than quickly, I guess would be. So basically, if you look here, this particular graph here kind of gives the idea. Your chronological, some people, the woman who is of the same chronological age, the tan one here, her biologic age is older than her chronologic age, compared to the blue one whose biologic age is younger than her chronologic age. But this is the general idea: is these clocks have been developed to try to predict biologic aging. And so here's a quote from the paper: post hoc analysis of 777 participants from the Do Health study on the effect of vitamin D. Omega-3, what was given was 1,000 milligrams a day, roughly, 660 EPA, 220 DHA, or what's called a simple home exercise program. And they looked at 4 next generation DNA methylation-based clocks: the PhenoAge, the GrimAge, the GrimAge2, and this one here, Dundon Pace. And they looked at the treatment only went on 3 years, and these people were already in their 60s, I believe, to begin with. Look over. These are the clocks that they use. These 3: PhenoAge clock, GrimAge2 clock, Dundon Pace clock. I don't know why they have these are all done. They all use different kind of algorithms to predict aging, but all 3 of them were used in this study. And here's what they found. For the PhenoAge, and if I make the point here, if point estimates, or the square, is to the left of 0, it means slower biologic aging than chronologic aging. Okay, so being over on the left-hand side is a good thing. And so we're looking at a change. Was there a difference in the biologic age versus the chronologic age based on this clock? And if you look at the omega-3 versus no omega-3, there is a statistically significant slowing of biological aging based on this particular clock in those who got the omega-3. How about the next clock? Here is what's called the GrimAge2 clock. Same thing. Using this different tool, omega-3 versus placebo, omega-3 treatment reduced biologic aging compared to chronologic aging. Next one, the Dundon Pace, whatever it's called. Same story, statistically significant reduction. And again, the omega-3 on this one was the only one that was statistically significant. And same for this clock, but not for this clock. On this clock, some of the other combinations were also significant. But the point is that omega-3 alone slowed DNA methylation clocks, PhenoAge, GrimAge2, and this other one, And all three treatments had added benefits to the pheno age. So vitamin D and exercise also helped. This, I think, is critically important right here. Overall, from baseline to year 3, the standardized effect ranged of omega-3 treatment from 2.9 to 3.8 months. Now, we've talked earlier about years of life added, but let's think about this. This is given for only 3 years. And if your aging clock slowed down by roughly 3 months in 3 years, then you could extrapolate, perhaps in 6 years, it would slow down by 6 months, in 12 years by a full year. So to have a roughly a 3-month slowing of biologic aging in just a 3-year randomized trial in older people is a really impressive finding in my view. In summary, our trial indicated the small protective effect of omega-3 treatment on slowing biologic aging over 3 years across several clocks, additive protective effects of omega-3, vitamin D, and exercise based on these tests. So that is really the primary story I want to get across. I did want to end with one complex slide. This is basically the mechanisms, a study that, it's not a study, but it suggested some of the mechanisms that we think about either fish intake or omega-3 supplementation doing. Anti-inflammatory markers are reduced by omega-3 and a reduction in pro-inflammatory markers. All these feed into a reduced overall oxidative stress and lipid peroxidation state throughout the body. There are antioxidant effects of omega-3, and there's direct action that we think on telomerase and telomere attrition. At the end of the day, it all ends up to be reduced telomere attrition, slowed aging. And that's what all this data really supports, that the omega-3s, when you have higher omega-3s for a long, long time, particularly even 3 years, but ideally for your whole life, that aging will just be slowed down and all the diseases of aging will be pushed further into your horizon. And so I want to end just with thanking my colleagues at the Fatty Acid Research Institute. There's some 17 of us here now, and we're working to advance the study of omega-3 study of all fatty acids, a pretty strong focus on omega-3 and health, and hope that has been helpful. I'll go ahead and stop sharing, and we'll turn it back to Tavis.
Doc, thank you so much. And I just realized I have something open. I was moving around while you were talking at one point because I had to get something to drink, and I was like, I forgot to close the back. So that's a little weird. Apologize about that. Let's dive into the questions. There's some really good questions in the Q&A, and let's just kind of talk about that. First question, is there any evidence connecting the omega-3 index with the health span as well as the lifespan?
Well, I think everything I just went over is essentially said the same thing. If you're staying alive now, I think the subtlety of the question is maybe you're staying alive, but you're staying alive longer sick. And I don't think we have evidence that that's the case. Particularly Heidi Lai's study looking at risk for unhealthy aging. They found the higher omega-3 levels associated with exactly that, a better health span, a longer health span. And so I think that paper probably be the best one to point out that, you're healthy longer, not you're just living longer.
Have you, is the information you provide today, have you ever written an article? Someone was asking, is there an article based on this? Because I know it's a lot of slides, a lot of information, but
It's a great idea. I should do it. No is the answer, but having put this talk together today, it would be a nice review article. I agree.
yeah, and so a little bit of a long question or a little bit of a long statement then to the question. In light of favorable phenotypic aging, also associated with plant-based precursor omega-3 in the diet, and with rising sustainability concerns in global marine EPA DHA supply, what role can the standard American diet rich pro-EPA terrestrial omega-3 sources play in inflammaging? And if you want to look at that question, you can always hit the question Q&A button.
Yeah. Oh, that's true. Yeah, no, I get the point. And the point is that kind of back to other sources of omega-3 and the whole, the concern about how in the long run, are we going to really out of our fish stocks, are we going to be able to generate enough omega-3 to to raise everybody's omega-3 index up to 8. That's seems unlikely. So there are a variety of other sources. And the one that's referred to in that question is, I think, probably called ahi flower oil, which is an oil that's from a plant that is richer than almost anything else in SDA, stearidonic acid, 18,4 omega-3, which is a good precursor to, it's a much better precursor for EPA than is ALA, the standard plant omega-3. So stearidonic acid in ahi flower oil is a potential new source, one of these novel sources of omega-3 that we can get from plants. And we mentioned the last time, there are algal products that produce EPA and DHA from essentially plants. And these are all going to be useful, I think, in the future to increase omega-3 status in our population.
Since Vasipa and I believe Lavazza are both ethyl esters, and the studies using these show an increase in longevity, what studies have been done with triglyceride forms?
Well, none in the same way. As a supplement, I think all the evidence that I've talked about from either the Force Coalition or the UK Biobank, or when they're talking about fish intake, when you're talking about fish intake or getting omega-3s from fish, you're primarily getting it from triglycerides and some phospholipids. So when the omega-3 levels are higher in those settings, it's from triglycerides primarily and some phospholipids. So I would assume it's the same thing. The ethyl esters are absorbed reasonably well if you take them with food. So if you see an effect with an ethyl ester, I'm sure you'd see an effect if anybody would ever do a study, long-term study with a triglyceride-based product. I'm sure we'd see the same thing. It's just that there's no financial incentive for anybody to undertake that study.
I think we'd see omega-3 levels go faster with triglyceride. And
yeah, should be. So,
let's see. Did you see any data related to RTG or triglyceride being more effective than other types of omega-3?
We've seen that in other settings, but not in this context of aging. Again, I think we know that the restructured TGs or just natural TGs are absorbed better than ethyl esters. You need a lower dose of the triglyceride form to raise the omega-3 index than you need an ethyl ester. I have no doubt that a triglyceride-based omega-3 product would be as, if not more effective than what we looked at here.
Excellent. Any other questions? We have just a couple more minutes. You can put them in the Q&A for Dr. Harris. Looks like another question came in. Is there any data about how low-quality rancid fish oil impacts health and longevity?
Yeah, there's some data. Well, not health and longevity, but health biomarkers of lipids and stress markers. And one really kind of outrageous study done from Norway that they took very rancid fish oil, very oxidized fish oil, intentionally oxidized fish oil. They put it in capsules, and they took the same oil that was not oxidized and put it in capsules, and they randomly gave them both to different groups of people for about, I think, a few months. And then they looked at the levels of lipids, the levels of oxidation markers, of peroxidation markers, et cetera, and couldn't find any difference at all between the two groups. So the body is pretty well designed to handle some fairly, I mean, none of the products you find on the market today would be considered nearly as rancid or oxidized as the stuff that was used in this study. So I don't worry much about that. I think it's far more important to think about the EPA and DHA content than to worry about the, how super pure the product is. That it's good, but I'm more interested in the EPA and DHA content.
This question says, not related to longevity necessarily, but is there any evidence of omega-3 reducing C-reactive protein levels?
Yeah, oh, sure. There's several studies. There's meta-analyses on that. That omega-3 treatment does lower CRP. Depends on how high it starts. And, if you got pretty normal CRP levels, it's not going to lower that. But if you start elevated CRP, it will lower it.
Excellent. Yeah, you would think by now a lot of the influencers or people just constantly focused all their attention on omega-6 seed oils would say, you know what, we need more omega-3 consumption. Only 7.8% of people take fish oil. And I don't know what that percent is that actually eat fish in the States, but you would think if we shifted our education to increasing omega-3, getting tested, getting more,
Right.
levels, we would see better outcomes in overall health. And
Right,
even the
right.
con-
Actually, I agree. I think that that distraction of the omega-3 or the seed oil is hurting us. That we need to spend the energy getting people to get more omega-3 and not worry about the omega-6 side of it.
Well, the Council of Responsible Nutrition published a big paper in 2022 looking at the cost savings of certain ingredients. Omega-3 was one, magnesium, vitamin D. The omega-3 potential was 40 billion in cost savings if we can just give everyone a gram and a half of omega-3 a day.
Wow.
By 2030, we'd save $40 billion in healthcare costs. So that's just something to think about.
That's great. Cool. Send me
No
the
more
paper.
questions? Nope. Go ahead,
I'd
Doc.
like to see that, Tavis.
Yeah, I'll send you that and we'll see you again. Doc, thanks again.
You bet. Bye.